Welcome back to the deep dive. We're diving into something pretty foundational today, the very fabric of modern medicine. We're looking at how we've defined diseases, how that's changing, and where we're heading with this idea of precision medicine.
That's right. And you've brought us some really rich source material excerpts from a detailed medical text. It really unpacks the history, the cutting-edge principles, and even touches on screening and prevention.
Yeah, this isn't just about, listing concepts. We want to get under the hood, see how the thinking in this field has evolved.
Exactly. The goal here with this material is to go deep enough that you feel like you're mastering these concepts, really seeing the connections, like the professionals do.
Right. Moving beyond just the surface level. Our mission today is to pull out those core ideas, follow the historical journey, and hopefully find those crucial insights hidden in the details. So, okay, let's unpack this and really aim for that level of mastery.
All right, let's start where it all began, or maybe more accurately, where the modern approach started taking shape. The way we defined illness for centuries, going way back to figures like Galen, was often holistic, but it lacked the kind of precision we expect today.
And the big shift, that came when? Late 19th century?
Yeah, around then.
Mhm.
And it was grounded in something called clinico-pathologic observation.
Clinical pathologic observation. Okay, what does that actually mean in practice?
Well, think of it like medical detective work, but with a really crucial final step. It was all about correlating what doctors observed while someone was alive, their signs, their symptoms,
Mhm.
with what they found after death during postmortem examinations.
Ah, okay. So you see the clues in the living patient, and then you examine the scene, so to speak, after they've passed to find the physical evidence of the disease.
Precisely. And because understanding the structural damage often meant looking inside, disease definitions naturally focused on the late stages, the kind of endpoints, often characterized by whichever organ seemed most obviously affected.
And this approach, it was really formalized, made systematic by Morgagni way back in 1761, wasn't it?
Absolutely foundational work. His meticulous comparison of clinical descriptions with findings from, I think it was over 600 autopsies at the University of Padua, that really showed the physical, the anatomical basis of disease.
He provided the structural why behind the clinical what.
Exactly. And this framework, it encouraged what's called inductive generalization.
Mm.
Basically, taking specific observations from lots of patients and applying them as general rules or definitions for a disease.
Okay.
And you combine that with Occam's razor, the idea of finding the simplest explanation, and it led to what we might call simplified disease definitions.
These Osslerian diagnostics, based on this approach, they were hugely successful for their time, right? Helped us understand and start treating many conditions.
No doubt, huge advances, but the source material is quite clear about the significant limitations.
And the major blind spot was,
It was its failure to distinguish the underlying causes.
Yeah.
The etiologies. You could have different root problems manifesting with very similar outward symptoms or structural changes, what the source calls common pathophenotypes.
right, different roads all leading to the same visible destination. Like, the text mentions multiple distinct causes that could all result in end-stage kidney disease or heart failure.
Exactly. On the surface, clinically, they might look very similar.
Okay.
And this is a key insight when you look at the evolution. As we've gained the tools to identify these deeper causes, our disease classifications have become vastly more refined. Think about things like neurodegenerative disorders or lymphomas.
Yeah.
Classifications that were once really broad are now highly segmented based on specific underlying mechanisms.
And that segmentation isn't just academic shuffling, right? It has real-world consequences for patients.
Oh, absolutely. Understanding the specific cause gives you much more predictable information about how a patient's disease is likely to progress, their prognosis. And crucially, it tells you which therapies are actually likely to work.
Right. Giving a drug that's effective for one cause of heart failure might be totally useless for another, even if the symptoms look identical on the surface.
Precisely. So, okay, we move from this powerful but limited observational approach based on anatomy. Then came the era with this incredible promise of molecular reductionism.
Right. The idea that surely every disease must have a single or maybe just a few molecular causes we can pinpoint.
That was the hope, wasn't it? The sort of idealized possibility driving so much research. Physician scientists focused intensely on molecular mechanisms, really believing they could break down every disease to its fundamental biochemical or genetic error.
And they did uncover a lot about how cells and molecules work in disease states. But here's one of the surprising insights from this material. Very few clinical manifestations could actually be explained by just one single molecular mechanism.
Yeah, it was a bit of a humbling disappointment for some. The source uses the sickle cell disease example beautifully, I think.
How?
Well, we know the exact genetic mutation, a single change in the beta globin gene. Simple, right? But knowing that doesn't actually predict whether a specific patient will suffer primarily from strokes or those agonizing pain crises or hemolytic anemia.
Mhm.
The same single molecular defect leads to this striking variety of clinical outcomes.
So we expected too much from oversimplifying things down to a single molecular cause. We didn't fully account for the, what does the text call it, extraordinary biological variety.
Exactly. That variety exists even in what we consider normal function, let alone how complex the molecular interactions underpinning disease actually are.
And the Human Genome Project, while obviously revolutionary in giving us the blueprint, initially kind of amplified this feeling, didn't it?
In a way, yes. Having the expanded genome didn't immediately reveal clear, simple, monogenic, oligogenic, or polygenic causes for most common diseases, as some might have hoped. I mean, beyond identifying some rare high-impact variants, of course.
The source material visualizes this, I think there's a figure reference, as an arc of progressive reductionism that sort of reached the plateau.
Right. It showed that just dissecting things down to the smallest molecular parts wasn't by itself sufficient to fully explain the complexity of how diseases actually show up and progress in real people.
It forced a realization that we needed a different paradigm.
A new path forward. And that path is described as a return to holism. But it's a fundamentally different kind of holism than the ancient approach. This is an integrative holism, meaning it takes into account the immense genomic context, environmental factors, other influences across multiple dimensions. It tries to see the whole interconnected picture.
And this complex, interconnected biological landscape, that's the territory of precision medicine.
Exactly. It means disease definition can no longer be broad and simplified. It has to become much more precise and ultimately much more individualized.
Right. If you think back to how traditional medicine was practiced, that observational method, coupled with the need to simplify things for diagnosis.
It naturally led to oversimplification of the phenotype, essentially grouping people based on similar outward appearances or symptoms.
Which, made sense for practical purposes at the time, didn't it? It simplified diagnosis and let doctors apply what they saw as specific treatments to those broad groups.
It did. And biomedicine, compared to fields like physics or chemistry, was sometimes viewed as less quantitative. Biological diversity was often treated almost like inconvenient noise you had to filter out. So the focus became distilling the observable complexity down to a core set of symptoms or signs that were reliably present across sick individuals. That system worked. I mean, it formed the bedrock of diagnosis and treatment well into the 21st century.
And we shouldn't downplay its successes. That approach led to incredible advancements, enabling large-scale drug development, treatments that saved and improved countless lives.
Absolutely true. But its limitations became increasingly obvious as we learned more. The source material points to significant predictive inaccuracies.
And maybe most striking, it mentions that large numbers of patients, the estimates go up to 60%, simply don't respond to the most effective drugs prescribed for their condition, as defined by those traditional broader categories.
So yeah, 60% non-response is a huge number. That really underscores the need for a more nuanced strategy if we want better outcomes for individuals.
Which brings us directly to the fundamental building blocks of precision medicine itself.
Right. And the first block is really refining how we describe and categorize patients moving beyond those broad strokes. Astute clinicians have always been aware of subtle differences they see, even between patients, given the exact same disease label.
These clinically apparent differences, that's what the source calls pathophenotypes.
That's the term, yes. A great example is heart failure. We now distinguish between heart failure with preserved ejection fraction versus heart failure with reduced ejection fraction.
HFrEF and HFpEF.
Exactly. Driven by technology like advanced echocardiography, we can now see these are fundamentally different conditions, even though both result in the symptoms of heart failure. And that distinction guides very different treatment strategies.
Okay, so pathophenotypes are the visible or measurable clinical differences. Then there's this concept of the endophenotype, or endotype. What's the distinction there?
Endophenotypes are characteristics of a disorder that aren't readily observable clinically, but are measurable internally. Think of distinguishing different types of vasculitis inflammation of blood vessels.
Okay.
You might distinguish them not just by the organs affected, but by specific blood tests. Serology is looking for autoantibodies or maybe immunophenotyping of cells. These markers aren't something you'd see just by looking at the patient, but they reveal a deeper underlying mechanism.
So endophenotypes are like the hidden, measurable traits that define a disease subtype, even if the outward pathophenotype looks similar.
Exactly. And these refinements, they're clearly driven by the clinical need, the need to improve prognosis and apply therapies with more precision, targeting that specific endotype.
It sounds like, in a way, experienced clinicians who really took the time to characterize their patients in incredible detail and then tailor therapies based on those individual features, they were already practicing a kind of personalized medicine.
They absolutely were using the tools and the understanding available to them at the time. But precision medicine aims to take this much, much further with new tools. And one concept here is orthogonal phenotyping.
Orthogonal phenotyping. Okay, that sounds technical. Break that down.
It basically means assessing clinical, molecular, imaging, or functional characteristics, endophenotypes, that might initially seem unrelated or orthogonal to the primary clinical problem the patient presents with.
Why would you look at seemingly unrelated things?
Because it can dramatically enhance our ability to distinguish subtle subtypes of disease. Diseases in one organ system might manifest very subtly in other systems, maybe revealing a different underlying issue that wasn't obvious.
Like a neurological disorder having subtle metabolic markers or maybe tiny structural changes somewhere else that aren't causing symptoms yet.
Exactly. Things that hint at a specific subtype or pathway being involved. So the argument here is that unbiased, comprehensive phenotyping, looking across multiple, maybe even unexpected systems, should really become the standard way we characterize patients in this new era.
Getting a truly multidimensional picture of the individual.
That's the goal. And to get that deep picture, you absolutely must couple that detailed phenotyping with comprehensive genomic profiling.
Right, and genomic profiling isn't just sequencing a little bit of DNA anymore, is it? The source lists quite a few different omics.
Oh yeah, it's layers upon layers now. Beyond just the DNA sequence, whether it's the exome or the whole genome, you're looking at gene expressions, so the mRNA being produced, then the proteins being made from that mRNA, the proteome, then the small molecule byproducts of cellular processes, the metabolome, how genes are turned on or off chemically, the epigenome, even modifications to the proteins after they're made, the post-translationally modified proteome, and, fascinatingly, the collection of microbes living in and on us, the metagenome, or microbiome.
Yeah,
Yeah.
that's a lot of data potential. But practically speaking, how much of that is truly available for routine clinical testing right now?
Well, many of these aren't routine yet, that's true, and a lot of what is available often comes from blood samples, which presents its own challenge.
How so?
While your DNA sequence is essentially the same in every cell, trying to get meaningful mRNA, protein, or metabolite profiles from a blood sample that accurately reflect what's happening deep inside, say, a failing kidney or a diseased brain, that's really difficult if you can't directly sample the affected organ.
Right.
Systemic markers like inflammation markers in the blood can be useful sometimes, but their correlation with what's happening specifically in an organ isn't always very strong.
Okay, so this is where the complexity really starts to build, but also where it gets incredibly interesting. The source points out something crucial. The functional consequences of a genetic variant aren't just about whether the gene is present or even expressed in a tissue.
No, it's critically dependent on the co-expression of its protein binding partners in that specific tissue.
Binding partners.
Yeah, the other proteins that it interacts with. Think of it like a team. A single player's action, a protein from a variant gene, only makes sense in the context of the rest of the team they're playing with on the field at that moment.
Forming specific functional pathways.
Exactly. These interacting partners form specific functional pathways or networks. And it's the dysfunction or the altered behavior of these entire pathways that actually drives the observable phenotype, the symptoms, and the characteristics of the disease.
So you absolutely need to see the whole network context, not just one isolated part, to understand the true impact.
Precisely. Even some investigational strategies, like creating induced pluripotent stem cells, iPSCs, from a patient and turning them into specific cell types in a dish, they have limitations because their gene expression patterns might not perfectly match the native cells in the body's complex environment.
And there's another layer of complexity mentioned: time.
Yes, time trajectories. A genomic profile is often just a single snapshot. But our gene expression patterns, our protein levels, our metabolites, and therefore our phenotypes, they change over time.
And these trajectories can differ wildly between patients, even those who seem to have the same overarching condition initially.
Absolutely. So ideally, repeatedly sampling and tracking these changes over time would be incredibly informative.
But I imagine that gets expensive pretty quickly.
It certainly can be, yes. Although the source does note that the cost of genomics, sequencing especially, is decreasing rapidly. So more frequent profiling might become more clinically feasible down the road.
Cancer seems like a field where they are already doing more frequent sampling with biopsies and liquid biopsies.
Cancer is definitely a prime example here, and it highlights both the huge promise and some really unique challenges of precision medicine. You can sample tumors repeatedly, tracking the evolution of somatic mutations, the mutations acquired by the cancer cells, and see how they affect known oncogenic pathways.
But cancer has its own set of hurdles, doesn't it? Like the fact that tumors change their genetic makeup so quickly.
Exactly.
Yeah.
That's a major one. The mutational landscape is constantly evolving, especially under the pressure of therapy, leading to the selection of resistant clones. Cells that happen to have mutations that let them survive the drug take over.
Which means targeting just one pathway with a single drug is often unlikely to achieve a lasting cure, because the cancer can find alternative pathways to achieve the same growth-promoting goals.
Right. And a third challenge is that tumors are often not uniform. There can be significant genomic heterogeneity within a single tumor. Different cells in the same tumor can have different mutations.
Which complicates targeting and just fuels that continuous evolution and resistance.
It does. So even with incredibly rich genomic data, interpreting it and applying it effectively in cancer precision therapy remains really challenging. It's often still semi-empirical, despite the data.
But the source does mention that combined therapies, maybe including immune modulators, still offer great promise.
Oh, absolutely. The strategy is moving towards combination therapies, hitting multiple targets or pathways simultaneously, much like we do in other fields. The source material even draws an interesting analogy here, comparing it to how we treat infectious diseases.
How so?
Well, in infections, standard practice is to identify the specific causative organism, the bug, and determine its sensitivity to various antimicrobials. That's precision treatment, right?
Right.
And we often use combinations of antimicrobials to prevent or overcome the development of resistance, just like the goal with combination therapies in cancer.
That makes sense. But even with infections, different people respond differently to the same bug and the same treatment.
They do, and that's where precision medicine principles come back in again. Individual differences, perhaps based on different endophenotypes, like the specifics of their immune or inflammatory response, mean that even treating an infection could potentially benefit from a more precise mechanistic understanding of the host, not just the pathogen.
So things like immune modulation for chronic infections could be a new frontier for personalized analysis.
Exactly. Understanding why one person's immune system handles an infection differently than another's, even with the same bug, is key.
Okay, so zooming out from the specifics, what does precision medicine need to truly evolve and become, standard practice across the board? What are the big requirements?
The source identifies several critical needs. First, we need even deeper, richer, more comprehensive personal phenotypic and genomic data. This is big data territory, requiring massive storage, complex analytics, and definitely advanced tools like machine learning and AI to extract meaningful patterns.
And we need data from normal populations too, right, to
Yeah.
establish baselines.
Crucial. We need that context to understand what constitutes a meaningful deviation. Second, phenotyping itself needs to become even more comprehensive and dimensionally richer.
Going beyond just the clinic and standard labs.
Way beyond, incorporating orthogonal data streams, data from personal devices, wearables, smartphones, detailed environmental exposure information, maybe even data about social network interactions, plus integrating data from the broader health system. The source has a figure showing all these inputs.
That sounds like a monumental data integration challenge.
It is. It requires huge investments in medical informatics to standardize, harmonize, and integrate all these disparate data streams into something coherent and usable.
Okay, and the third need. You mentioned AI and machine learning.
Right. This feels like maybe the biggest challenge. Figuring out the minimal data set needed to reliably predict a meaningful outcome or response to therapy.
Separating the signal from the noise.
Exactly. It's one thing to collect petabytes of data on every person. It's another to analyze it effectively, to identify what's truly predictive, eliminate redundancy, correctly weight the most important factors, and ideally find easier to collect signatures or biomarkers that are practical and cost-effective in a real-world clinical setting.
And that's where machine learning and AI are absolutely essential to find those complex patterns that humans just can't see in that volume of data.
Precisely. They're the tools we need to navigate that complexity.
So, circling back to a question we touched on earlier, if complete knowledge of every single molecule isn't possible or even necessary, how precise does precision medicine actually need to be?
The source argues, quite pragmatically, I think, that it needs to be sufficiently predictive to be practical and cost-effective in clinical care.
Hmm.
You don't need perfect knowledge of every molecule in a person's body to significantly increase the probability of choosing the right therapy for that individual compared to the traditional approach.
It makes me think of concepts from clinical trial design, like the difference between prognostic and predictive enrichment.
That's a great connection the source makes, too. Prognostic enrichment is about selecting trial participants based on factors known to affect their likely outcome regardless of treatment, maybe like disease stage, just to make the trial group less heterogeneous.
Okay.
Predictive enrichment, which is really central to precision medicine trials, uses detailed data analysis like genomic profiles or specific biomarkers to predict who within that group is most likely to respond to the specific treatment being tested.
So, precision medicine isn't about achieving perfect absolute predictability, but about making predictions accurate enough to genuinely guide better clinical decisions for individuals. The source says its boundaries are, quote, imprecise, strictly speaking, but sufficiently predictive.
That's a really key takeaway. It's about fundamentally shifting the odds in favor of the individual, based on a deeper understanding of their unique biology.
And these powerful principles of precision medicine, they aren't just for treating diseases that have already developed. They're increasingly being applied to screening and prevention, too.
Absolutely. The goal shifts here. It becomes identifying individuals who are at significantly higher risk of developing a particular disease down the line so we can intervene proactively.
And this is crucial because some interventions, maybe they carry risks or burdens themselves.
Exactly. Interventions that might be too high risk or too costly or just too inconvenient for the general population could be highly beneficial, even essential for someone identified as being at much higher risk. It changes the benefit to harm calculation.
So, what kinds of data are key for predicting risk in people who are currently asymptomatic? Germline genomic data seems like the most obvious starting point.
It's certainly a big one and probably the most advanced right now. We know about mutations in high penetrance genes like BRCA1 and BRCA2 for breast and ovarian cancer risk. Finding those has very clear implications for preventive care.
Like recommending prophylactic surgery, oophorectomy, or mastectomy, or initiating specific intensive screening protocols like MRI scans.
Right, things that carry their own risks and wouldn't be appropriate for average risk individuals, but where the potential benefit dramatically outweighs the risk for someone with that high genetic predisposition.
And it's not just those rare, high-impact genes anymore, is it? The contribution of rarer, moderate penetrance variants and even combinations of common low penetrance variants is being recognized.
Definitely, which leads to the development of polygenic risk scores, or PRSs. These attempt to combine information from potentially hundreds or thousands of genetic variants across the genome to estimate risk for common, complex conditions, like cardiovascular disease.
And as the cost of sequencing continues to drop whole exome, whole genome sequencing, this kind of deep genomic risk assessment, seems likely to become much more widespread in preventive care, doesn't it?
It seems inevitable, yes. The information potential is huge, though interpreting it accurately and translating it into actionable advice is still evolving.
Are other omic data types being explored for risk prediction too? Proteomics, metabolomics?
They are being explored, yes. Proteomics and metabolomics could potentially offer more dynamic insights into someone's current biological state, maybe reflecting recent exposures or pre-disease changes. But creating clinically reliable predictive markers from them has proven challenging so far.
But maybe things like circulating tumor DNA.
Ah, yes. Analyzing fragments of DNA shed from tumors into the bloodstream, or looking for molecular markers in stool samples, those approaches show really exciting promise for detecting certain cancers, like colorectal cancer, at a very early asymptomatic stage, non-invasively. That's a very active area.
And imaging data too, right? That's being integrated more for risk stratification.
Definitely. Things like coronary artery calcium scoring, using CT scans to help refine risk assessment for cardiovascular disease and guide decisions about statin therapy, or even using features from screening images like mammograms to predict the likelihood of potential harms, like a false positive result, which could help tailor screening frequency or follow-up for individuals.
It makes perfect sense that screening and prevention are gaining so much importance in this precision era. We have new imaging technologies letting us detect changes much earlier, maybe even at the cellular level.
And our growing understanding of the molecular pathways that drive disease initiation offers the potential for chemoprevention, using specific drugs or agents to actually prevent the disease from starting in high-risk individuals.
Which ultimately, preventing disease or catching it incredibly early is the ideal outcome, right? Both for individual health and potentially for saving significant healthcare costs down the line.
Absolutely. And the source material even briefly grounds this modern approach in the basic principles of screening that were established decades ago by the World Health Organization back in 1968, often applied initially to common disorders with a large public health burden.
So it's not replacing those fundamental public health principles.
No, not at all. It's providing powerful new tools and incredibly granular data to refine those principles, making screening and prevention potentially much more targeted, more effective, and more personalized.
Wow. Okay, so we've covered a huge amount of ground in this deep dive. We've journeyed really centuries of medical thought, from defining diseases based mostly on what we could see after death.
Through that intense period focused on breaking everything down to the smallest molecular parts, understanding both the incredible promise and the inherent limits of pure reductionism.
We arrive at this modern integrative approach, an approach that really embraces the complexity of biology.
Right. Precision medicine, as we've unpacked it, clearly isn't just about finding a single culprit gene. It's about integrating these incredible layers of complex, multidimensional data: the clinical picture, the pathophenotype, the hidden internal markers, the endophenotype, the full spectrum of genomics.
And how those genes and proteins interact in complex networks, plus factoring in environmental exposures, lifestyle, maybe even the microbiome, all to understand the unique biological state, that reticulotype, as the source called it, of each individual person.
And as we aimed for that level of mastery today, we saw the incredible potential that stems from that deeper understanding. The ability to understand an individual's disease with far more precision, predict how it might progress much more accurately.
Choose therapies with a much higher likelihood of actually working for that person. Maybe even finding new uses for existing drugs by understanding those network connections.
And extending those same powerful principles to identify who really needs intensified screening or specific preventative interventions before disease even strikes.
But as you said, the journey is far from over. The challenges are immense.
Definitely. The sheer volume and complexity of the data we can now generate is staggering. Making sense of it all requires incredibly sophisticated AI and machine learning tools that are still being developed and refined.
Plus the dynamic nature of diseases like cancer, that constant evolution and resistance.
And the ongoing challenge of figuring out how much data is truly enough to be clinically useful and also cost-effective in a real-world healthcare system. These are huge hurdles still.
So, thinking about this whole journey, this shift from defining people by broad medical labels to understanding them at this level of exquisite individual biological detail, how might your own personal understanding of health and disease shift?
Yeah. What does it really mean when your health isn't just categorized alongside millions of others based on a few symptoms, but potentially understood as this unique, dynamic constellation of your specific biological makeup interacting with your specific environment and history?
What does that imply for how you might think about prevention strategies you could consider, or treatments you might receive down the line, or maybe just your own personal health journey moving forward in this rapidly evolving landscape?
It's definitely something to keep pondering as this field continues its incredibly rapid evolution.